So we're going to do reproduction today and Wednesday. Today and Monday. Remember, exam on Wednesday. I'm going to send an email out with the review session times on Monday and Tuesday night. But Monday and Tuesday night, review session at 7. And I'll send you the room locations and post them as well. Okay, so we've been talking about the endocrine system. And having done the endocrine system and having done the nervous system, I now want you thinking about what are the ways those two combine to get input from the world, sensory input, input from the body, and then control something really complex, reproduction, that involves inputs from multiple locations that all have to be coordinated in time. Behavior needs to be coordinated among different organs. Physiology needs to be coordinated, and that all needs to be connected to what's going on in the real world. So we're going to do that looking at reproduction, and specifically mammalian reproduction. I hope you have reviewed or will be reviewing some of the basic things about reproduction, asexual versus sexual reproduction, and so on. That's old material. Where I want to start talking about is sexual reproduction, which occurs in all animal phyla. At least all the ones in which we know anything about reproduction. It also occurs in protists, and undoubtedly the earliest animals inherited the key features of sexual reproduction, especially meiosis and fertilization, from single-celled organisms. So a lot of the stuff we're talking about, again, uses the same molecular toolkit we've been talking about all along. So if we have a sperm that is swimming along toward an egg, it's got a flagellum, and in a moment we'll focus in on some of the parts of a sperm. It reaches an egg and binds, and you wind up with the sperm nucleus a one-end sperm nucleus inside that egg. It is really important that, first of all, that the egg only allows in sperm of the right species. You don't want sperm to be coming in from an incompatible species. Otherwise, that egg is basically wasted. You're going to wind up with infertility or an organism that has such an odd combination of genes that it can't survive. You also need to prevent more than one nucleus getting in, because if more than one sperm nucleus gets in, you wind up with a triploid or tetraploid or whatever organism, and now you have a developing zygote with the wrong number of chromosomes, often lethal. If it's a triploid, pretty much lethal. If it's a tetraploid, rarely successful, but sometimes there are some tetraploid organisms, but for the most part it doesn't work. Okay? So how does that happen? So if we zoomed in on a part of that membrane, and we looked at a time span of about 20 to 30 seconds, from the top to the bottom of this figure, we can see the events. Sorry, the resolution isn't great on all of these. One key point is that in a particular species, selection favors sperm proteins on the surface that will bind to egg proteins on the surface of the egg that have to match with a lock and key fit. You need a precise match between sperm proteins and egg proteins. These are egg recognition proteins or sperm recognition proteins. So that the wrong species comes along, it just bounces off. Okay? For a marine organism in particular, the world is loaded with sperm. They're all over the place. Not in really high densities, but they're all over the place. When you swim in the ocean, you are swimming in a diluted sea of sperm. Okay? You really don't want those to fertilize any eggs unless they're the proper match to a species. So that's what bindin is. So bindin is a protein, highly variable across different species, specific to that particular species, and it's expressed on the egg, on the ovum. And the sperm has egg recognition proteins, those black dots, that mean it'll only bind to the proper species. It's not very helpful for a sperm to bind to the wrong species either. So strong selection for unique binding. Okay, so binding happens. Something that was noticed early on in eggs were that there were these little roundish things called cortical granules. Can you read that okay? Cortical granules. Cortex is just a root word. It refers to the outer part of something. Okay, so the cortex of the brain is the outer part of your brain. Cortex of the kidney is the outer part of the kidney. Cortex of a cell is the outer part of a cell. Okay. It's a terrible word because every time I look at it, I think apple core. Core is the middle. It's a horrible word to use for the outer part of something. I think one may be a Latin work and Latin root and the other one Greek, but if it is, they got them horribly mixed up. Anyway, cortical granules. So there are all these things right underneath the membrane. And when you look at the events of fertilization under a microscope, what you see is that shortly after binding happens, these cortical granules fuse with the membrane. What makes that happen? Well, what makes that happen is that these recognition proteins, when they bind, that opens a calcium channel. I don't know if it's a metabotropic or an ionotropic receptor, but upon binding, calcium channels are opened. That opens more calcium-activated calcium channels. So when binding happens, you get calcium coming in and activating more and more calcium coming in. So there's a wave of calcium coming in. And if you have a vesicle and the right proteins associated with it, what have we talked about in terms of neurotransmitter release or hormone release from vesicles? Can you make that happen with calcium? Okay, so calcium binds to a calcium binding protein on these vesicles. And I can't zoom in anymore, but calcium binds on these. They fuse with the membranes, and their contents get to the outside of the membrane. Okay, or at least outside of this inner membrane. So you get this area that has the contents of the cortical granules, and if there's water present, those cortical granules, the contents of those cortical granules expand tremendously into a gel. What would you guess the cortical granules are made of? What's inside them? Okay, gags. Okay. So what you're getting is cortical granules with gags or similar molecules that are really hydrophilic. And so these gags take up a huge amount of water, and you get swelling. That swelling forms something that's called a zona pellucida. It's not so critical that you know the name, but I do want you to realize upon fertilization, you get this coat of a jelly-like material that additional sperm can't get through. Okay, so you get a thick gel layer of gags, and any other sperm that comes along just gets stuck in it or bounces off. As part of that process, any remaining bindins get removed. So within 30 seconds or so, those remaining bindins are already being removed, such that any sperm that do happen to get through that gel-like layer can't do anything. Okay, so there's a physical barrier really that happens really quickly, and then a barrier that happens because the egg recognition proteins, the sperm egg recognition proteins, no longer there. And the sperm nucleus is now inside. Gil?
So the purpose of the calcium is to destroy the membrane of just the granule, right? And then whatever's in the granule comes out and expands.
So the purpose of the question is the purpose of the calcium to destroy the granule, release the granule is the terminology, so release those granules. So the purpose of the calcium is, so if we imagine there's an ovum up here, okay, and I am a bindin. All right, can you picture that? Okay, so along comes a sperm, giant sperm is coming down through the ceiling, it hits me on the head and it sticks to my head. Okay? Then I'm activated, I open calcium channels. Calcium comes in and we get a wave of calcium moving all the way along the surface, and at every cortical granule the calcium binds to a calcium binding protein. We release gags, so all around you and spreading out over you are these gags. Water, any water molecule nearby gets caught, so these things expand tremendously with all those gags. And the cortical granules contain them, so the purpose of that wave of calcium is to release the cortical granules and form this barrier. Calcium as a second messenger is also activating processes that remove the bindin from the membrane. So, right, calcium doesn't need to do just one thing. If a calcium binding protein can activate something that cleaves off a part of bindin, then you'll release the bindins. Okay, make sense? Okay. So some version of this happens across animals. Most animals have sperm that have let me do a new sheet here. So, sperm that have an acrosome an acrosome with enzymes. Upon binding, those enzymes get released, and they help to digest their way through debris and things around the outside of that egg. Egg recognition proteins. They're the ones that are going to bind to bindin. And a large part of it is the nucleus, and it's haploid, 1N. That's the head of the sperm, or the head piece. Then there's a tail. Within it is a flagellum, and mitochondria. The mitochondria are producing the ATP that allows that flagellum to keep swimming, to keep beating. And then there's a projecting end piece that's the last part of the flagellum sticking out. Okay. Okay, so if we're thinking about the events that are producing sperm, there are testes. Testes are the organs that produce sperm. Okay, so testis in a human, ovoid, typical in mammals and birds, lots of species, but they don't have to be. What's critical is that within the testis are these tubules that are containing stem cells and developing sperm cells. So the sperm are developing in these tubules, and around the edge are 2N stem cells. Professor Wawrcik's lab here studies these in Drosophila. And the stem cells go through meiosis to eventually develop 1N sperm. Those move out, so typically smooth muscle contractions along here move out mature sperm and release them for fertilization. So spermatozoa. When we talk about mammalian reproduction, or vertebrate reproduction in general, if we imagined a giant testis up front, okay, another one of these lectures I love. Okay, imagine a giant testis up front, and let's cut it in half. Okay, this part always makes me squirm a bit. Okay, let's cut it right down the middle. And what we're going to do is we're going to cut all those tubules, right? So when we cut it down the middle, and look at it in cross-section, we're going to have a whole bunch of tubes that we've cut, actually many more than this. What we're going to care about here is that there are cells in between these tubules, interstitial cells. Interstitial is just a generic term for in between other things, in the interstices. Okay, so interstitial. In mammals, they're called Leydig cells, named after the person who first described them, Leydig. That's not so critical, but interstitial cells will work just fine. I would like you to recognize the term Leydig cells if you see it. And these are the cells that are producing testosterone. So these cells have LH receptors. From the video, okay, they have LH receptors, and that induces the production of testosterone. Testosterone is necessary for spermatogenesis. You need some, and you also need FSH. So if we looked at these stem cells, we need FSH to bind to receptors on them to trigger the production of sperm. So FSH, follicle stimulating hormone, was first discovered in females, so it actually got named for what it does in females, but it actually affects both sexes, and it causes gametogenesis. Okay, so Leydig cells LH, and LH causes sex steroid secretion. Testosterone and estrogen. Okay, so that's what's going on in the testis. Similar things going on in the ovary. We'll come back to that. How is this regulated? So first we need to get our animal to be adult. So I've drawn a brain. I've drawn a pituitary gland far larger than it really is because we need the space. There are GnRH neurons. And at puberty, sexual maturation, you start getting an increase in GnRH release. Before then, almost no GnRH secretion. Reproductive system is inactive. Okay, if you've got a little brother and you want him to mature early, at say 4 years old, just start giving him GnRH injections. Okay, and that will lead to maturation. Don't do that. So, GnRH receptors. So GnRH receptors in the anterior pituitary bind that GnRH. There's a pituitary portal blood system that carries the blood. I already talked about it briefly. It's also in the video. And these cells in the pituitary release FSH and LH. Both of them are protein hormones, so they both bind to surface receptors. And between the two, we get hormones that stimulate maturity. So those are the hormones, the sex hormones. And they're doing things like triggering changes in muscle development, changes in fat deposition, changes in hair growth, changes in the brain, so changes in, they're activating neuronal populations that are part of the behaviors and the regulatory systems for fertility and reproduction. Gil?
Are there two sets of cells that produce LH and FSH, and this is just simplified?
Yeah, no, this is a weird one. It's one type of cell. So usually one cell type, one hormone. That's the most common. That's what you'd guess. But in this particular case, one cell type, two hormones. And the detail is really not important to us, but that happens to be how it is.
What about in the ovary? Did you have those leucite and the other cells that seem to...
So in the ovary, we're going to come back to that. In the ovary, there's two classes of cells, one of which are, you can think of them as nurse cells. They're feeding the ovum, and they're responding to FSH by putting more and more nutrients, more and more stores, and so on into the ovum. And then a separate set of cells that are responding to LH and producing estrogen. Okay, so this is going to lead to physical maturation, behavioral maturation, and gamete production. In a male, that gets us to fertility. Okay, in a male, there's no need for any kind of cycle. So simpler system to think about. Many animals are seasonal, so they suppress this during part of the year, and what they do is they suppress GnRH neurons. So if you simply inhibit GnRH neurons, you get less GnRH release, less LH and FSH, fertility drops. In a human, you can do it with really extreme conditions. So starvation, for example, will shut down fertility. A level of starvation that is actually fairly severe will stop GnRH production, and a male will become infertile. Get the resources back again, and fertility will return. Now in females, it gets more complicated, and in females, it's both really interesting, at least to me, hopefully to you guys too, at the end of all of this. In a female, we get this very complicated dance of neurons, hormones, and communication back and forth between these different cell types and different organs. So that's what we're going to talk about next. It's what's in the video. So I'm going to run through this today, and then we'll do some problem solving and go back over a few more things on Monday. Okay, so we have, actually let me show a couple slides first. I want to remind you of a few things about reproduction. So we are amniotes. When the embryo develops, it has both the embryo proper, what's in purple here, actually what's in, let me color it in myself. Here's the embryo proper in the purple I just added. And then we have a yolk sac. Okay, so there's a yolk sac, which in us doesn't seem to do much. It's there, but it doesn't seem to do much. We have an amnion. Okay, there's an amniotic cavity. This is the amnion. So same embryonic membranes we already talked about. And then the allantois and the chorion, they form all this other tissue. Let me try a different color here. So here's the chorion. Okay, in the egg of a bird or a fish, the chorion cell layer that forms around the whole outside of the yolk, the embryo, everything. And then the allantois, here, which grows out and mixes with it. And in a mammal, you have a chorioallantoic placenta. Both the chorion and the allantois together form the placenta. The chorion, the outermost layer, going all the way around that embryo and everything else. And the allantois coming out, carrying the blood vessels out from the embryo to be very close to the maternal tissues. Okay. So maternal blood, vessels here are very close to the blood vessels that will be coming out from the embryo. And those vessels are close enough to make efficient transfer of nutrients and oxygen in one direction and waste molecules, carbon dioxide, ammonia, and so on in the other direction. Okay, so how do we make that happen? Here's some of what will make more sense to you once we come back to this later. So this is part of a summary, and then this is another summary sheet you can use for reference, but this is not a way to learn it. Trying to learn this by memorizing these just as they are won't work. You want to break it down into pieces and have the pieces make sense. Okay, so we're going to start with the brain, and I need a big enough brain to put some cells in here. Going to need a big pituitary. Here's the anterior pituitary. I've separated off. We're going to need the hypothalamus. We're going to start with GnRH neurons. GnRH neurons are releasing their GnRH into the pituitary portal system. That's binding to GnRH receptors, and we get LH and FSH into the blood. We're going to need a big ovary. And we're going to start on the right with a follicle that's inactive. The follicle is the cell that will become the ovum. It's in partway through meiosis, and it's surrounded by some of these little support cells. Which have, some of them, FSH receptors. And others have LH receptors. Okay, the ones with FSH receptors bind FSH, and those cells can be thought of as nurse cells, and they increase the mass of the ovum. They also trigger it to complete meiosis. So as this process is going on, it completes meiosis until it's a final one-end cell. So over time, so the nurse cells, the ones I'm talking about as nurse cells, their function is to put materials into the ovum. And they have FSH receptors. The LH receptors are on sex steroid secreting cells. Okay? So the nurse cells are feeding resources into that ovum. The ovum starts off tiny, and it grows to be the biggest cell a human will ever produce. Sorry, only in women. Men can't do it. Okay? So it's going to be a huge cell. Those resources have to come from somewhere. They're being fed into it by these nurse cells. So the nurse cells are moving materials into there. That word is inactive. We'll try again. They start off inactive. Okay, they start off inactive, but once they start getting LH and FSH, that activates them. So this word is activate. Okay, so it gets bigger. You get more of all of these cells. They're actually in layers, but I'm not going to bother with showing the different layers. Nurse cells are on the inside, so we'll just put some around there. We get more cells and more FSH, more nutrients going into the ovum. Okay, so here's our FSH receptors. And more LH, at least at the beginning, there's LH that is still fairly abundant, and we get estrogen. Okay? Now we have more cells, right? We have more cells. So are we going to have the same amount of estrogen or more estrogen? Okay, so we get more estrogen, which I usually draw by making the E larger. Okay, more estrogen here. But there's another wrinkle to this. Kiss cells, I'm going to call them, these are kisspeptin neurons. They got the name because they were discovered by a group in Pennsylvania, in Hershey, Pennsylvania. where Hershey's Kisses are made. They regulate fertility, so they named them Kiss, after Hershey's Kisses. Kiss peptin, peptin for peptide. Okay, so these are Kiss cells, and they have estrogen receptors. Remember, estrogen crosses cell membranes. So if we looked at the inside of these Kiss cells, we'd have estrogen coming in, binding to its receptor, moving into the nucleus, and regulating the kisspeptin gene. And in these cells, the receptor binds to a repressor. Okay? So we get less kisspeptin. Kisspeptin causes action potentials in GnRH neurons. So the kisspeptin receptor opens sodium channels. We get action potentials in GnRH neurons. So what is estrogen going to do to GnRH secretion? Okay, so estrogen is going to inhibit GnRH secretion, right? Estrogen is inhibiting the production of kisspeptin, so we're going to have less kisspeptin, which means fewer action potentials per second in GnRH neurons. Make sense? Okay. So estrogen negative feedback so we get less GnRH. We're going to have more, the same amount or less LH and FSH. Okay, so you're going to get less LH and FSH. So coming down to our growing follicle here, we have less LH, less FSH, but more cells. Okay? So even though there's less FSH and less LH, we still get even more estrogen. Okay, does that make sense? Okay, so we're reducing GnRH and LH and FSH, but because there's more and more cells on this particular follicle, we're still getting more estrogen produced. Simeon? Okay, so I may be guessing what you're wondering about. So this seems pointless. Okay, why is this follicle, which depends on getting LH and FSH, why is it inhibiting LH and FSH? It needs the LH and FSH to grow. The reason it's doing this is because this ovary may have 100,000, a million ova in it. You do not want all of them to grow at once. Imagine a million tuplets. Okay? You don't want them all to grow at once. So what's happening is that only the largest follicles get enough FSH to keep growing. In other words, by this point, those inactive ones, the ones that never got started, they never will get started. There's not enough FSH and LH to get them going. Of the ones that got started, there may have been 100 that started growing. But as they grow, there's less and less LH and FSH. And only the largest wind up getting enough FSH and LH to keep growing. And the others fail, and they die. It's a sad story for most of those follicles.
How does it keep growing if there's not enough FSH?
Okay, so here's the balance. How does it keep growing if there's not enough? There's only enough if you have enough of those cells with FSH receptors. So it's not how much per cell, it's the total amount you get from all the cells. So a follicle that only has a handful of cells doesn't get enough. But a follicle that has hundreds of cells, even though each cell isn't producing as much, isn't doing as much, they're getting enough. Okay, so it's the aggregate number of cells that's what matters. And the ones that are smaller don't get enough. Let's say one of them dies. So let's say the biggest one dies, then you'll have slightly less estrogen, and the next biggest one now is getting enough LH and FSH to take over. But if that biggest one never dies, then the second biggest one in a human, normally, will not get enough FSH and LH, and it will fail. And you wind up, usually, with just one offspring. What might you do to increase the odds of having twins? What would you inject people with? Okay, you could inject them with GnRH to get more LH and FSH. What else? Okay, FSH directly. Okay, so, and that's actually, for fertility treatments, that's one of the first things we try. It's one of the cheaper, still not that cheap, but one of the cheaper things we try is just GnRH injections. Gil?
Wait, but isn't twins is when the egg splits after it's been fertilized, right? So,
Sorry, it's...
don't twins form when the egg...
Don't twins form when the egg splits after fertilization. So that's identical twins. And in identical twins, you only had one ovum, but after fertilization, they split, separated, and you wind up with both of them growing and maturing. That would be identical twins. Fraternal twins, which could be boy, girl, boy, boy, genetically different, no more similar to each other than any other pair of siblings. Okay, they happen when you have two ova, both fertilized. Okay, two follicles, two ova, and then you get non-identical twins. Probably a few of you in here are parts of non-identical twins. Okay, maybe some identical twins as well in here. But okay, let's move on. Not relevant. Okay, and there was there another question? Did I get? Okay. Okay, so what we have now, we have a system that is going to work to get these follicles growing. It's going to work to keep maintaining the biggest one or biggest ones. If you're an animal that, like a sheep that normally has twins, okay, then it's typically the two biggest. There's enough LH and FSH secreted typically for the two biggest. And for some animals that have, like mice, that have 6 or 8 or 10 offspring at a time, there's enough FSH and LH for 6 or 8 or 10 follicles to make it, okay? Okay, so now we have a system that will grow a bigger and bigger ovum, but is that the goal? An ever bigger ovum that eventually gets so big that mom is really just an ovary with a giant ovum in it and all parasitically sucking all of her nutrients out? We want to do something with it, right? We need to release it and get it fertilized. So as this process is going on, there's more and more estrogen because there are more and more and more of these cells. Less LH, but more cells. And now we need something that is going to trigger the events of ovulation and trigger behaviors related to mating. In most species, mating happens only around the time of ovulation. Humans are weird in that mating happens at any time in the cycle. And for the most part, humans are unaware of when ovulation happens, when somebody is actually fertile. Okay, so what happens at this stage is that high estrogen interacts with another population of cells in a different location that I'm going to call KISS-positive cells, or if you want, KISS-positive feedback cells. And in these, again, these also have estrogen receptors. And kisspeptin gene. There's the coding region. And again, it binds but to an enhancer region. Okay, so now in these cells, binds to an enhancer region. And so in these cells, the more estrogen the more kisspeptin released. So once you get a high enough level of estrogen, the KISS-negative cells are not releasing kisspeptin. They're doing nothing at this point. They're really inhibited. But the KISS-positive cells, which don't have all that many estrogen receptors, so you need really high levels of estrogen to activate them, just not enough receptors in there. But when you get those high levels, they start secreting large amounts of kisspeptin. And you wind up with a positive feedback loop. Kiss positive, oops, cause more GnRH, which cause more LH, which cause more estrogen, which cause more kisspeptin. Okay? So at this stage, we've now switched from estrogen inhibiting GnRH to estrogen stimulating it. So LH levels go through the roof. And those high LH levels bind to receptors in the wall of the ovary and break up the extracellular matrix. So high LH breaks the extracellular matrix. So if our giant egg here, fertilized, sorry, not yet fertilized, or giant egg here, now that high amount of LH has broken up the connections between all of these cells that were holding it in the ovary, and out it pops, ovulation. So high LH, which only happens, remember, if that follicle is really big and producing a huge amount of estrogen. That's the signal that it's mature, the amount of estrogen it's producing. That causes ovulation. Okay, and it also triggers the high LH and the surge in estrogen, triggers mating behaviors in females. In most species, that's what triggers mating. And so as this ovum is moving down the oviduct, it meets sperm coming the other way, and we get fertilization. And the events we talked about at the beginning of class. Here's our developing embryo. And it has binding proteins so binding proteins that make it stick to the wall of the uterus. And the estrogen, the increasing estrogen, has triggered growth of cells and so the uterine lining has gotten thicker and thicker, ready for the embryo. Okay? Okay, so we've taken the reproductive system all the way from inactive, prepubertal or infertile, all the way down to producing an egg, getting it fertilized, and having it attached to the uterine wall. The uterus has been prepared. Next time we'll talk about events of pregnancy and we'll work a few problems.